Good afternoon, and welcome to the American Floral Endowment's Grow Pro webinar series. I'm your moderator, Michael Wojcicka, and I am the head grower for Bell Nursery in Springfield, Ohio. I oversee and manage the production of a wide variety of ornamentals for hundreds of stores across the country. Today's session is on identification and management of root diseases in floriculture crops. On behalf of the endowment, I'm excited to be part of AFE's Grow Pro webinar series that features a new topic every month presented by an industry expert. The webinars are free to everyone thanks to the generous support of AFE sponsors. The views and opinions expressed in these webinars are those of the speakers and do not necessarily reflect the views or positions of AFE. As an independent nonprofit, AFE cannot endorse any specific product or opinion. Today's webinar is sponsored by Bioworks and Fenstop Fungicide, a product of Gowan. Bioworks helps customers in the horticulture and specialty agriculture markets successfully deliver crops to market with biologically based solutions and support. Fenstop Fungicide, a product of Gowan Company, is a highly effective fungicide against downy mildew, pythium, phytophthora, and foliar phytophthora in greenhouses. If you'd like to learn more about our sponsors or are interested in becoming a sponsor for a topic, you can find that information on AFE's website at endowment.org/growpro. Today's session was pre-recorded in English by Dr. Francesca Hand. After the presentation, Dr. Hand will join us for a Q&A. Feel free to submit your questions through the Q&A feature or chat at any time. We'll answer as many as we can before the end of the hour. Unanswered questions may be answered through a separate email exchange. The session is being recorded and will be shared to AFE's YouTube account. Through YouTube's accessibility features, you can access closed captions in other languages. To get started, I'd like to share a bit about today's speaker. Dr. Francesca Hand is a professor of plant pathology and state extension specialist for ornamental plant pathology at The Ohio State University. Previously, she received a PhD in plant pathology from the University of Florence, Italy, and she conducted postdoctoral research at the University of California, Davis. The purpose of her research and extension program at OSU is to investigate disease epidemiology, biology, and ecology of fungal, oomycete, and bacterial pathogens, from which she develops detection and control strategies. Dr. Hand, welcome and thank you for presenting today on root diseases in floriculture crops.
Well, thank you for the opportunity to talk with you today. My name is Francesca Hand, and I'm a professor of plant pathology at The Ohio State University. And I will be talking to you about identification and management of root diseases in floriculture crops. So as you all know, because you work in the industry, this is a very, very comprehensive industry. It is valued at almost $6.7 billion. This is as reflected in the latest summary of floriculture crops that was done in 2022 and published in 2023. And floriculture crops are include a very wide variety of crops. And what I will be talking about today pretty much applies to all of this. So we can have issues with plant diseases on annual bedding plants, such as begonias and petunias, impatiens, and things like that, through herbaceous perennials like dahlias and peonies, chrysanthemum, and then all the way to some woody perennials that are grown for the beauty of their blooms. And these includes, of course, roses, gardenias, hydrangeas, and crops of that sort. So as I said, many of these crops share some common enemies, and these are the diseases that I will be talking about today, which are categorized as root rot diseases. These are diseases that are caused by soilborne pathogens. Soilborne pathogens are pathogens that naturally reside in the soil and then can attack plants from the soil or the substrate, and really include the two big groups of organisms, the oomycetes and the true fungi. The oomycetes are also known as water molds, and they are really very different from fungi. They once were thought to be one group of organisms that we now know that they're two very separate groups of organisms. Among the oomycetes are pathogens such as pythium, Globisporangium, which is just a newly renamed group of organisms that used to belong to the Pythium genus. There is Phytophthora species, and then a relatively new genus known as Phytopythium that has been recently reclassified from organisms that share some characteristic between Pythium and Phytophthora. Among the fungi that cause root rot diseases are Berkeloiomyces versicolor, which is used to be known as Telaviopsis versicolor. Again, it's been recently renamed, but it's the same pathogen. And then Rhizoctonia solani. And so today I will be touching on all of these different pathogens. Taken together, so not a specific organism, but taken together, these soil-borne pathogens that cause root rot diseases are the largest cause of plant loss in floriculture production. And that is because once the pathogen finds that root system in a susceptible plant, it starts invading that plant and killing that root system. And when a root cell is dead, right, it's necrotic, can no longer generate new roots. And so that plant remains small, or all the way to, it can die depending on the size of that root system. In general, if you have a young plant that has a small root system, that can very quickly lead to a dead plant. Whereas if you have a more adult plant with a more robust root system, depending on how many sites of that root system are under attack at one given time, you may see a variety of symptoms that vary from debilitation, compromised crop uniformity, all the way to plant death in severe cases. The most common root rot pathogen in the greenhouse is Pythium. And when I say Pythium, I include in this genus also the recently reclassified Globisporangium. So Globisporangium and Pythium both cause a disease known as Pythium root rot, and it is the most common root rot in the greenhouse. It is the primary cause of dumping off in seedlings, but it can also be found in more adult plants and potted plants beyond the seedling stage. Different species of Pythium and Globisporangium have different temperature requirements, which can really tell us when we could expect to find one species or the other, or what environment within our production system may be more susceptible or prone to infection by one species or the other. In general, Globisporangium irregulare and Aphanidermathium are more warm season or warm temperature pathogens versus Globisporangium ultimum. It prefers more cooler environments, and so it may be more of a problem in the winter or in cooler areas of the production pipeline. Globisporangium ultimum and Pythium Aphanidermathium are the two most aggressive among the Pythium species, and then Globisporangium irregulare is more associated with stressed plants, so can cause disease when a plant is already stressed. So just to understand what happens at the below ground, right, in that root zone, is that the pathogen finds that root, starts attacking that root, and as it kills that root, it releases toxins and other type of compounds that make the cell become necrotic. And so if you look at a plant that is under attack by root rot pathogen, the root system really looks discolored. And this is very much evident if you compare a healthy plant with an infected plant, like you see here on the left. You have a very discolored root system. You barely see any roots, and then on the right side you have a healthy white root system. One important diagnostic feature with Pythium root rot is this thinning of the lower end of the stem right at the insertion of the root system. As you can see, these were snapdragon plants that were grown in a cut flower farm, and you can see this thinning here of the stem right at the insertion of the root system, and that is often characteristic of Pythium. Another characteristic diagnostic feature of root rot pathogens is the ability to create these mushy rot, and I'm referring specifically to Pythium. And when this happens, the outside layer of the root becomes very mushy, as I said, and sloughs off really easily if you pull your hands through that root system. And when that happens, you're pretty much removing the outside layer of that root system, leaving just the central part of that root attached to the plant. And so we call this symptom rat tail, because it really looks like the tail of a rat. And so it's something to look to if you pull up a plant that is showing symptoms of root rot, and then just inspect that root systems and pull those roots to see if you observe any of that rat tailing. Now, when about 20 to 30% of the root system is compromised because of the activity of this pathogen, that's when you start seeing symptoms in the above ground part of the plant. So when you see symptoms above the ground, the part of that root system is already compromised. And because the roles that roots have in a plant, right? They absorb nutrient, they absorb water. If the root system is compromised, the plant isn't able to carry out these functions in a regular way. And so what you may be observing above ground is symptoms of nutrient deficiency, for example, right? So the plant isn't able to uptake the nutrients, and so is now showing symptoms of nutrient deficiency. You have an example here where you see this chlorotic foliage. Now, that may lead you to think that you need to to pump up your fertilizer, but really it's not. The problem is not that there isn't enough nutrients. There aren't enough nutrients in the soil. Is that the root system isn't able to uptake those nutrients because the root system is compromised by the pathogen. So foliar discoloration or nutrient deficiency symptoms are very common when the root system isn't working. So is the uniformity of the crop. So you may have plants that look much smaller compared to others that aren't affected. You may see wilting associated with plants because the plant isn't able to uptake water the same way it isn't able to uptake nutrients. You can see that plants start to drop their leaves, and then in extreme cases, you can see plant mortality. Here you have some other examples of these symptoms that I just talked about. You can see here to the left a healthy poinsettia, and then a one that is definitely stunted compared to the one on the left, and that's the one that is affected by Pythium at the root level. And then here you see this impatiens crop, again, with some plants that are much, much smaller. They haven't really, pushed through the plague stage. And then you have others that look much bigger that were planted at the same time. One common phenomenon that is observed in SAM plants, sometimes this is observed in poinsettia production, is that in periods of the day in which the plant is transpiring at a higher rate, let's say in the middle of the day when it's very hot, the plant is showing wilting because it's losing water at a much higher rate than it is able to uptake it from the ground. And so during those moments of peak transpirational loss, you see that the plant is wilting. And then when the temperature cools down and the plant doesn't transpire as much, then you see that it recovers. And it's something that, may get overlooked, thinking that it's just, water availability, but really is that the root system is not functioning properly. And so it's not able to keep up with the water requirement of that plant in moments of peak transpirational loss. Now, once a plant is infected with Pythium, but also other oomycetes, the pathogen will produce a specific type of spore that is considered the resting spore, and this spore is called oospore. If you look at this section of this root right here you see in the center is the center part of the root that is called the vascular cylinder, and then you see these round-shaped structures right here, those are called oospores. And again, they're the resting spore of Pythium that the pathogen produces at the end of the infection process. Once the root system has been colonized by this pathogen, these spores form, and these spores are the main way of for this pathogen to stick around, right, in production. These spores can decompose, the root system that is affected by Pythium can decompose in the substrate, and then these spores are released in the substrate. They may be picked up by water, and then they can stick around. And once they find a susceptible plant, they can grow towards this plant to infect it. As a matter of fact, these spores are built to sense the presence of a plant. When a plant is planted in a pot or is planted in a field, it normally and naturally produces some compounds that it releases around the root system. While the pathogen is actually able to sense the presence of these compounds produced by the plant. And when it senses them, it also senses a gradient, right? So the closer you get to the plant, the higher the concentration of these compounds will be in the substrate. And so when it senses that gradient, it grows towards that gradient and then eventually finds the root system and is able to infect it again. Now, Pythium is a very persistent pathogen. So once it's in the greenhouse, it's really hard to get rid of it. And so it is important to understand how it can be introduced in the greenhouse and try and avoid introduction in the first place. Contaminated plant material such as plugs or cuttings is definitely a way in for any pathogen. But in the case of oomycetes, recycled irrigation water is also very important in continuing to reintroduce the pathogen into production. Anything that comes in contact with the plant, such as contaminated media, debris on floor that can be picked up by splashing irrigation, as well as tools and pots and benches that can come in contact with the plant. Once the pathogen has reached that plant, then development of disease, so infection of that plant, is favored by conditions such as soggy media, poor drainage of media, too much water, and then high populations of fungus gnats. So from a cultural control standpoint, there are a few things that is important that are done to prevent introduction and to prevent disease development, and that includes not overwatering plants. We will see when we talk about Phytophthora that there are some spores that are produced by these oomycete pathogens that are able to swim in a film of water. And so providing the plant with that extra water just makes it easier for those spores to go around. Watch for continually soggy media, because that's an indicator of something may be going on, or you may have favorable conditions for that plant. Select media that are more porous and well-draining. It's also important to test the soluble salts level weekly to ensure a healthy root system. If you have conductivity level above 1.5, usually that puts the plant more at risk of creating micro wounds. Those high salt concentrations can cause wounds in the root system, and in any wound is an open door for a pathogen to get access to the plant. So it's important to avoid that. For crops that are irrigated with overhead irrigation methods, such as hosing, it's important to keep those hoses off the ground, because a hose that touches the ground can pick up debris that may contain spores or mycelium of Pythium, and then the application of water to the crop can just disperse that inoculum. Containers from plants that have been confirmed to be infected with pythium should not be recycled, and also fungus gnats populations along with algae in the greenhouse should really be monitored and controlled. And this is because the two really go side by side often. Fungus gnats feed on algae. Algae can form very easily in the greenhouse, especially along irrigation lines where there's maybe accumulation of fertilizer, and the population can increase very, very quickly when there's so much food available. And so monitoring and controlling fungus gnats is important also to prevent introduction and spread of pythium within the greenhouse. Now, from a different set of tools, so outside of cultural control methods, there are both chemical and biological control methods available for pythium root rot, and you have listed here to the left fungicides that are effective for pythium, as well as biological control products that are effective for pythium. Please keep in mind that these tables aren't all inclusive. There's some other products that aren't listed here, and then also consider that anytime you decide to apply a fungicide, you should really try and incorporate this fungicide into a rotation. And what that means is that you should be selecting across different FRAC codes. FRAC stands for Fungicide Resistance Action Committee, which has developed this numerical system to make it really easy for any grower to identify the mode of action of that fungicide. Any fungicide that share the same number in their classification hits the pathogen the same way. Doesn't matter what is their trade name, they hit the pathogen the same way. And if you keep hitting the pathogen with the same mechanism over and over and over again, that pathogen will eventually learn how to bypass that effect and will develop resistance to that fungicide, making that fungicide no longer useful and no longer effective, right? This is the case of mefenoxam, which is a very effective active ingredient for oomycetes, but unfortunately, there's records of increased insensitivity to this active ingredient, especially in pythium species that were isolated from floriculture crops. So choice of these fungicide is really not recommended as a first measure. It can be incorporated into a rotation. It can be tank mixed with other active ingredient to make sure that resistance development is decreased in chance, and also to make sure that even if you have an insensitive pythium in the greenhouse, that maybe there is something else that is hitting it at the same time, so you still see effect. As far as biological control options is concerned, it is very important to understand some basic principles of using biocontrol. Biocontrol can be very effective, but it must be used at the right time. It must be used before the disease pressure builds up, okay? So it's important to be used at sowing, it's important to be used at transplant, but if there's already high disease pressure, that product is very unlikely to succeed in managing that problem. So often we recommend applying a biocontrol agent after a chemical fungicide, so the chemical fungicide may maybe knock down the population of that pathogen, increasing the chances of success for the biocontrol agent. Another important thing to consider is that many biocontrol options, not all, but many, are real, living organisms. And as any living organism, they are influenced by a lot of things. One of them is the food that they find, right? So fertilizer regimes can influence these organisms. The plant itself can influence the organism, the type of pathogen, or sometimes the species of the pathogen can influence the efficacy of these organisms. So it's very important with biocontrol to set up some trials to understand what works best in one's production conditions. Another oomycete root rot that is not as commonly found in the greenhouse is Phytophthora. This is less common than pythium, but usually when it's found is more aggressive, so can still cause a variety of problems. There are 5 main species of Phytophthora that are found on floriculture crops. The most prevalent is Phytophthora nicotianae, but there is also species such as Drexlerii, Cryptogia, Tropicalis, and Palmivora that are associated with many floriculture crops in the United States. Often we see more Phytophthora problems in cut flower production where crops are grown directly in the soil, but that doesn't mean that we can't find Phytophthora in the greenhouse, just less likely. Depending on what host is attacked, we can have root rots, so the disease at the root level. We can have crown rots, so the root system is not necessarily fully compromised, but most of the activity of the pathogen is at the crown level. And then sometimes the spores of the pathogen can be splashed, dispersed onto the foliage, and we can have foliar blights that are caused by Phytophthora, which is not something that you would necessarily think that Phytophthora can do, but it's actually quite common. Here is an example of the extensive damage that Phytophthora root rot can cause in crops that are grown directly in field soil, such as you see this ranunculus crop over here, where it was completely devastated by this Phytophthora infection in high tunnel. The symptoms below ground and above ground are very similar to what we've already looked at for Pythium. So again, you have a healthy root system to the left here, and you have a extensively necrotic root system to the right, where you can barely see any roots because they're all brown, and so are easily confused with the color of the substrate. A root system that looks like that clearly cannot uptake nutrients, cannot uptake water, and so above ground you will see similar symptoms that we've talked about before, which include a variety of nutrient deficiency symptoms, but also wilting. And then here you see an example of crown rot, where the plant rots from the crown, and then the foliage just dies off from that point of infection. Now, both Phytophthora and Pythium produce a type of spore type of structure that is called sporangium. And a sporangium has different shapes and sizes, and that's what we use in a laboratory to identify the specific organism that we may be finding on an infected plant. But the important information about this structure, again called sporangium, is that it contains sometimes a very high number of tiny little spores called zoospores that have the ability to swim in water. So these sporangia are basically containers, they're sacs that at some point open up and release these zoospores that are able to swim in a film of water. And when that happens, the zoospores are very happily spreading in the greenhouse, especially if a plant is overwatered, if there's maybe floating conditions and things like that. If those zoospores get splashed, dispersed onto the foliage, then that's when we start seeing those symptoms of blight. And a blight is just a very rapid necrosis of the foliage that starts from an initial infection of the zoospores and then progresses onto the rest of the foliage. Now, from a control standpoint, biocontrol is really not yet there as far as efficacy towards Phytophthora. So the majority of our tools are going to be in the chemical side of things. Many of the fungicides that are listed in the table that I shared earlier about Pythium can be used for Phytophthora as well. But with Phytophthora, we have an additional series of fungicides that are listed here that do not work with Pythium. So it's very important to know which one of these two you have because, they work with Pythium but don't work for Phytophthora. So it's very important to know which one of the two you have because you need to select the appropriate fungicide to make sure that it's effective for the specific pathogen that you have. Again, this is not an extensive list, but it does include a variety of fungicides in different FRAC families. Again, look for that number to identify a rotation. There is a fungicide, one of the newest molecules that has been tested for efficacy against Phytophthora is effective, but also has the plus of providing really long-term control. So can, persist in the plant for a longer period of time and can be reapplied with a longer application time frame. All right, moving on to non-oomycete root rots. We're going to talk about two root rots that are caused by fungi. And the first one is a disease known as black root rot. This disease is caused by a fungus that was previously known as Telaviopsis basicola, but has been recently renamed as Berkeleiomyces basicola. It is the same pathogen, and you will be understood if you've learned it to be Telaviopsis and think that it's Telaviopsis, because, as I said, it's the same pathogen. The pathogen has a very wide host range and can affect a variety of floriculture crops, mainly bedding plants in the greenhouse industry, but it can infect all the way to adult trees. So it's a pathogen that has a very wide host range. The name of this pathogen is very important to identify. It identifies a characteristic environmental requirement for this pathogen, and that is an alkaline environment. As you notice here, pH conditions that are above 6.2, so an alkaline environment, are very conducive to disease development. And if you pay attention to the name of the fungus, the basicola part of it, it indicates basic, right? Basic or alkaline. So it really indicates that a requirement for that pH to be above 6.2. So that's a very important thing to keep in mind as we talk about management in a minute. High soil moisture content, high fungus gnat populations, and reused flats from previously seed crops are also very important for disease introduction and spread. It is a root rot, so the first thing you will notice is probably that there are some plants that have irregular stand, some plants are full size, some plants are much smaller, and that is often noticeable in flats where the plants were seeded all at the same time. The above ground symptoms will vary from chlorosis to purpling, sometimes a combination of chlorosis and purpling. And then once you observe the root system, you will be able to see pretty much nothing. So the root system may be very, very small and very, very dark, okay, very, very black. And that is the reason why this disease is known as black root rot. Okay, the rot isn't mushy like with PTM. It's not the root system doesn't slough off because it's not mushy, but the root system will be very much underdeveloped and very black in its size, especially if the plants are infected from the time of transplant. The pathogen can move into the crown of the plant if from root infections, and if you observe closely a root system that is infected with the pathogen, you will be able to see these areas of dark black lesions, and this is where the pathogen accumulates its reproductive structures, which are called chlamydospores. These spores are formed very effectively by the fungus, and they are very good at sticking to plastic surfaces. So often they stick to the plastic of the pot or the flats or the tray, and that's how this pathogen can move around in the greenhouse. From a cultural control standpoint, given what I just talked about, the ability of these chlamydospores to stick to plastic is recommended not to recycle containers if a crop was found to be infected by black root rot. If choosing to reuse these containers, it's very important to sanitize them thoroughly, and that includes first using high pressure water jets and then following with a peroxide-based disinfectant. Sometimes that can be coupled also with a dip in a 10% diluted household bleach. Many of the Thelabiopsis infection or black root rot infections come from incoming plugs. And so it is very important to inspect the health of the root system when you get incoming plugs, because once you've introduced those infected plants into the greenhouse, it's very, very hard to get rid of that pathogen. Because of what I said regarding the preference for the alkaline pH, it's very important to manage the pH and make sure that it's below 6.0 if possible. And then because of the preference for high humidity in the soil, it's important to use a well-draining potting mix, not overwater, and again, manage fungus gnats because they can carry the pathogen by picking it up from the root system while they're feeding on that root system. There are a variety of fungicides available. The ones included in FRAC 1 are the most effective and really have impressive performance against this pathogen in many research trials over the years. So they're a very good product. And then partners that can be used in a rotation with FRAC 1 include triflumizole, fludeoxonil, and polyoxin D zinc salt. And finally, the last root rot that we'll talk about is Rhizoctonia. Rhizoctonia is a basidiomycete, so it's a fungus, and it's a pathogen that differs a little bit from everything else that we've talked about because it prefers warm, moist soils that have high oxygen levels. So the more porous the substrate is, the more problems with Rhizoctonia one may have. So it's a little different from what I just said with all the other pathogens. We refer to this pathogen as an agent of Rhizoctonia diseases. This is not really a root pathogen. It's a pathogen that attacks the plant at the soil line and can attack the plants at the foliage, but really doesn't cause root rots. So it causes stem rots and foliar blights, especially in plants that have very tight canopy or are grown packed into flats. So here you see the characteristic symptom of Rhizoctonia infection, which is the presence of this sunken canker at the base of the stem. And that canker can be very extensive and can expand to girdle the entire stem. And so when that canker has girdled the stem, then the plant collapses on the surface of the media. And you can see here these healthy herbs on the left, and then you can see these seedlings that are just like collapsed on the surface of that pot because of the presence of that canker at the base of the stem. And that can be a very big problem. Here you can see plants that have, with all the stems that are collapsed. But one important thing to remember is that regardless of the presence of that canker, if the leaves of a plant touch the substrate and there is Rhizoctonia on that substrate, the pathogen can move onto the leaves and colonize and necrotize the leaves without causing that stem canker. And we see these especially in environments with high humidity, which are very much conducive to that foliage blight. And when the pathogen attacks the foliage, then kills that foliage, and you can see that it forms these web-like threads, which are part of the body of the fungus. It's called mycelium, that can hold the dead tissue together. And you can see here, they look almost like a spider web. Those are these web-like threads called mycelium or hyphae that keep together these tender plant tissue that is all blighted and necrotic. So from a disease management standpoint, it's important to keep field soil out of the propagation area. Field soil is very much a reservoir of Rhizoctonia. and so can be a source of inoculum into production. If a plant is found to be infected, it should be removed from the production area, and the greenhouse should be cleaned of any debris. And then there are a variety of fungicides that are available. They should be applied preventatively to avoid having the disease in the first place. And again, there is a variety of options for rotation across different FRAC codes. So I only have a couple slides left here, and I really want to emphasize the importance of proper diagnosis. As you can see, there are a lot of different pathogens that can cause similar symptoms, especially if one only looks at what you can see from above ground and you really don't inspect the root system. But the organisms that can cause these diseases are very different, and not everything that works for one organism can work for all of them. So it's very important to have a proper diagnosis and seek diagnosis from a reputable laboratory. There's plenty of laboratories across the country. There are university-based or non-university-based that can really do a good job at diagnosing the disease. For them to be able to do that, it's very important that you know what to send them to make sure that the outcome of the diagnosis is the best possible. That includes, whenever possible, do not send just a leaf or a stem or a piece of the plant, but collect a whole plant and send the whole plant whenever that is possible. If you have plants that are starting to show symptoms and plants that are completely dead, it is important to send a variety of sample, a set of plants that shows a variety of symptoms, and likely plants that contain the healthy tissue along with the dead tissue, because if we receive a plant with just dead tissue, we really aren't able to do much with it in the laboratory. It's important to wrap the sample in a paper towel, put it in the fridge until you're ready to ship, and then package your sample carefully to make sure that it doesn't get damaged during shipping. Here's an example of things not to do, is to put plants, and they're beautiful inside their pots, but then the media isn't really separated from the foliage, and during shipping, the media can go all over the place, risking contamination of the tissue that is now going to be very difficult to work with. So something like this would be much better, to wrap that root system and the soil so that it doesn't touch the foliage. And then finally, it's very important to include sample documentation with as much detail as possible. If you submit to a diagnostic lab, there's likely going to be a form that you have to fill out. And it's important to include all the information that is required in that form, because we're really playing detective when we get these samples, and any little clue can help us through the diagnostic process. Attach pictures if you have them. Pack the plants in a rigid container and possibly ship using next day delivery service to make sure that the sample gets to the lab fresh and doesn't sit in a warehouse over a weekend. And with that, I would like to acknowledge how important both the American Floral Endowment and the Association of Specialty Cut Flower Growers have been in building my research program and supporting my research program in diagnostics for floriculture crops. So I would like to thank them, along with the Ohio Department of Agriculture and the Ohio State University. And I would also like to thank BioWorks and FenStop for sponsoring this session today. Thank you. And I will leave you with an invitation to attend the next Grow Pro webinar, which is going to be on Tuesday, June 18th. And it will be focused on the use of drones in cut flower production by Dr. Nansen from UC Davis. And I will be happy to take any questions you may have.
All right, Dr. Ann, thank you for the presentation. We're going to go ahead and roll into the Q&A part of the presentation now. So I do have a question here. I know you touched on it in your presentation, but Lori Weiss was asking, in relation to not using plastic pots and flats that are sanitizing agents, are there any sanitizing agents that would be effective against oomycetes or true fungi?
Yeah, so there was a slide, I think slightly after the question was typed that had instructions on what products can be used. And just to reiterate, is sanitizing with high pressure jets and then using some peroxide-based disinfectant. Some studies have shown that followed by also a 10-minute dip in diluted household bleach is effective.
Thank you. And then I have a question from Allison Perez asking if it's possible to isolate in vitro all these pathogens.
Not all pathogens can be isolated. Some pathogens are called obligate parasites. They cannot be taken out of the plants. They need to be on the plant, but none of those are root rot pathogens. If we're thinking about like powdery mildew or downy mildew, those are pathogens that we can't isolate in vitro. So we need to make our observations directly on the plant. But as far as root rot pathogens, they're all culturable.
All right. And here we go. I have one from Carlos. Is it generally a good practice to tank mix fungicides when the identity of the pathogen is unknown? That's a good question.
Would not recommend anybody to apply fungicide without knowing what the problem is. I know that, I've talked about the importance of diagnostics. If a sample is in good conditions and is received by a lab that knows what they're doing, it will be just a quick turnaround to let you know what the problem is. And I would make recommendations based on, what the problem is rather than not knowing.
Awesome. And then another good follow-up question here. What can somebody do while they're waiting for their results to come in from a lab?
Yeah, so it is important to isolate those plants from the rest of the plants that aren't looking diseased. So, we used to call this quarantining. It's not going to be as long as 40 days, but definitely if you see something that looks suspicious, it is important to remove those plants from the production. And I always say, even the plants that are immediately adjacent to the ones that are showing symptoms, they may not show symptoms yet, but are likely to be infected already. And so it would be important to separate those from the rest of the production.
Awesome. All right. And then here's some more. So for someone who doesn't typically send in lab samples for diagnostics, and if they're not getting an immediate answer, is there a way that can be improved and get an answer quicker?
Yeah, so I wish, this was an easy answer. Some pathogens, we have very quick tests that we can perform right when we get them in the lab. I make an example for oomycete root rots. We have immunostrips, which are these very quick strip tests, like everybody I'm sure is familiar with, in-home COVID tests nowadays. And so they work pretty much the same way. And so those are very quick tests. We receive a sample, we test the root system. A grower may even have those in-house and do the testing themselves. And those are, very quick turnaround in a matter of minutes. So the information can be, sent to the grower immediately. In other cases, it really isn't doable, and we have to be able to extract the pathogen from the plant. So, it's like you get a blood test and then you have to wait for the results of that blood test before a doctor can prescribe anything for your health. And it's unfortunately, it's the same thing in plant diagnostics.
Awesome. And so Karen actually touched on something that you mentioned in her question here. Do you recommend products like Agdia Immunostrip for Phytophthora testings or other in-house testing options?
Yeah, absolutely. I mean, the Agdia immunostrips are very, very useful. There's a variety of them, not just for root rots. There's many of them for viral diseases, bacterial diseases, etc. They are a very good, tool to have in production, especially for big operations where there's a lot of material, all the time. I definitely recommend them. I do want to say, it would be good to still send a sample in for diagnostics if you have a positive or a faint positive result, because sometimes there can be, like false positives or samples that may not have like a bright, line that shows a positive result, then you may need to do some follow-ups.
Awesome. So another follow-up question here. So if I have a root rot disease in production, why does it really matter what the specific disease is?
Yeah, I mean, it's very important to know, right? It kind of goes back to the question that was asked earlier about, tank mixing fungicides, not knowing what the problem is you want to use something that you know is effective. And let's assume that, you have a fungus or an oomycete. We have shown that not all products that have activity against fungi also work for oomycete and vice versa. So it's very important to know, to know what you have before you apply a fungicide. And that also goes back to trying to limit development of resistance in pathogen populations. Anytime you apply a fungicide, that pathogen is going to be hit with that mode of action, and then you reapply it again, and then you reapply it again. Eventually, the fungicide will stop working. And historically speaking, pathogens have developed resistance to all kinds of things over the years. So it may happen, quickly.
So I know you mentioned fungicide resistance from efinoxam and seeing some of that out there. Are there any other fungicides that might be showing some resistance against Pythium, or Pythium that
So
might be showing resistance against fungicides?
yeah, so Adorn, so fluopicolide is another one that is recommended to be tank mixed. I am not aware of other specific active ingredients at the moment that have been reported with decreased sensitivity against Pythium, but I would expect things that are used extensively to eventually lead to that outcome.
And then I got another question here from Juan. What is your experience in control of Fusarium? I'm not going to try to say the subname.
Proliferum.
I'll leave
I
it.
actually do not have experience working with Fusarium, so I cannot answer that question. I can say, generally speaking, that Fusarium is a very, very complicated beast. It is very hard to control, and there are limited chemical options that are effective also. So as a general rule of thumb, it is one of the hardest to control plant pathogens.
I think that wraps it up on our questions. A lot of people are saying thanks, and I agree. This was a wonderful presentation, and I'm looking forward to bringing it back to our team here.
Great.
Oh, got
Thank you.
one more question here, and then we'll go ahead and start wrapping up. What methods do you recommend for resistance analysis?
Yeah, so some labs offer, screening pathogens for a series of active ingredients. For the most part, this is done in research labs. So I would recommend getting in contact with the ornamental pathologist in the state where you are, and inquire whether they're offering this service. Some programs do, and I think it's a really important thing that within a state we are aware of what is the status of fungicide sensitivity or insensitivity thereof.
And I think that goes ahead and wraps us up. I know there is a question in there about asking if the slides for the presentation will be available. I know one of our AFB team members is going ahead and answering something in there quick. But I do know we post all the recordings online.
Yep, so I know these will be available on the website.
And we'll go one last question here. This is a good question too. So I'm going to go ahead and ask. Little late coming to the presentation, but I caught your comments regarding PAA and bleach, but did not hear anything about quaternary ammonia products. Do you have any thoughts regarding those?
I'm not familiar with if there is a difference specifically with quaternary ammonia products as opposed to peroxide, hydrogen peroxide. I'm not aware of whether one is more effective than the other.
Awesome. All right. So we will go ahead and wrap up. Thank you so much. I know I appreciated the presentation, and so did a lot of our attendees. Want to thank all of our attendees as well for joining us today for another session of AFE's Grow Pro webinar series. And welcome you to join us next month for Dr. Christian Nansen's presentation on the use of drones in cut flower production on Tuesday, June 18th at 1 p.m. Eastern Time. You can register at endowment.org/growpro, And while you're there, check out our past webinar recordings, other grower-related resources, and research reports available to you for free thanks to industry support. If you find this webinar series and AFE's other industry resources valuable to your business, please consider making a donation to AFE's 2024 annual fundraising campaign. This campaign kicked off last week and will run from now until June 28th. More information about this year's campaign and sponsorship information can be found at endowment.org/rise2024. We ask that you please complete the brief survey about today's session where you can suggest additional topics and help us continue to improve these webinars. Thank you again for joining us and have a wonderful day.